Hi, I'm Meredith Blackwell. I'm here at IMC, International Mycological Congress number 12 in Maastricht, Netherlands. And it is the 13th of August, 2024. And we're here to talk to people. And would you introduce yourself?
Yes, my name is Pepijn Kooij. I'm originally Dutch, but I've been outside of the country for about 15 years now and currently live in Brazil.
Where in Brazil? It's a big country.
Yes, I live in the state of Sao Paulo, in the city of Campinas, but I do my work in Rio Claro.
Okay, and where is that in relation to where you live?
It's about like an hour more land inwards even.
Okay, and you were here in the Netherlands, ever?
Yes.
Okay.
I did my.
You were born here.
Yes, I was born here, and I did my bachelor's and master's degree here in the Netherlands in Wageningen. After that, I moved to Denmark to do my PhD.
Who'd you work with there?
With Koos Boomsma,
Okay.
evolutionary biologist working on ants, where I was a little bit the odd person out because I used to work with the fungal side of fungus growing ants, whereas all the others worked on the ants themselves.
Yes.
And after that I moved to Kew Gardens in London, where I worked for 5 years. The same topic, but more into the phylogenetics and the start of genomics part.
Who was director when you were at Kew?
When I arrived, it was Kathy Willis.
Okay.
But during my stay, that changed to Alexander.
Okay, I was just curious. I used to go to Kew a lot. Arthur Bell, who was a previous director, was the director then, who had taught me at Texas, University of Texas,
Nice.
and then returned home. Yeah, but Kew is a wonderful place. And
Yes,
must
beautiful.
be nice to work.
Yes.
Yeah,
Yes.
but no ants there. No fungus growing
ants
ants.
there.
Okay.
No.
Okay.
But I really focused on the fungal story of fungus growing ants. Because one of the things that we don't know, what is the closest free living fungal relative of the fungus that the ants grow. And they have the largest collection of dried mushrooms. So I went down in the fungarium collecting everything related that I could find to build a gigantic phylogeny and see where it ends.
Now, I once worked with someone who was working with some of these ants in Minnesota. And so I'm wondering which group of fungi are they growing? Are the Lepiota relatives or what?
So there's
The
different
mushrooms,
groups.
yeah.
It started all with Leucocoprinae.
Right.
And that involved in several different groups within that tribe of the family Agaricaceae. But there was one single genus of ants that started growing something completely different. Nowadays it's called Myrmecopterula, so in the Pteralaceae.
Okay.
So that's very different, of course.
And see, I found some members of the Pteralaceae in
Fantastic.
Louisiana, and as endophytes. And so they let me on the paper to talk about this and the fact that they were endophytes. That might be where the ants picked it up at first as
Fantastic.
endophytes.
Yeah.
That was a long time ago. I'd forgotten about it. Yeah.
Yes.
But very interesting. So you were in Denmark,
Yes.
and then what happened?
Well, I finished my PhD, and I stayed for a postdoc for about a year to wrap up the ideas, and then I found a position in Kew Gardens, where I stayed for 5 years as a early career researcher.
Yeah. And was Martin there at the time?
Yes, Martin Binatolno was there.
Yeah.
Martin Ainsworth.
Yeah.
Paul Kirk, I met him there as well.
Yeah.
So all the big mycologists from the UK, they were still there. So it was a great time to meet all those people, and I learned lots.
Yeah, and so then how did you get to Brazil?
So I was towards the end of my contract, and it was difficult to actually get a permanent position within Kew because they're limited. And then I during my stay, I actually met my colleague André Rodriguez, who's standing there. And he works on the antagonistic fungi of the fungus grown by ants.
Okay. And what is that genus?
There's several genera, but the most known one is Ascovopsis.
Okay.
Often
That's the
seen,
one I've heard of.
yes,
Yeah.
often seen as a parasite, but within our group, we are starting to think that maybe it's more opportunistic, depending on which species we're talking about.
Okay.
But we started talking, and he would like to have somebody with more experience on the mutualistic fungus. So then there was an opportunity of a grant for improving internationalization in the university there. We applied for that, and I was able to move. The grant initially was for 1 year, with the possibility of an extension for 1 year, but it was during the pandemic, and they had troubles with getting people for that, so they asked, do you want 2 years? You just need to say yes and you'll get 2 years. Okay, so I
No
had
proposal to write.
Exactly, so I was there for 3 years with this grant, and while I was there, I wrote a grant looking at the asexuality of this fungus, of the mutualist. It is not per se asexual because it can grow mushrooms, but the ants maintain it without mushroom. Because I think that's much more beneficial for the mutualism as a whole. So I wrote a grant for that, and I got that funded. And I started 1.5 years ago, and it's a grant for 5 years in total.
Oh, okay.
Doing lots of genomics, even single nucleus genomics I'm trying to implement there. So laser dissecting single nucleus to try and sequence those.
Okay, so how old is your clone that you're looking at? Are they really clonal?
They are for sure clonal. How old? I have no idea yet. We don't know enough yet of these fungi to really draw these kind of conclusions. But we're getting closer and closer.
Well, it's a really, really interesting system to work with. And
Yes.
we have a few. I lived in Louisiana in the United States, and so we had one that was called the town ant that got up. Do you know that term, town
No,
ant?
no.
Yeah, because they built cities, I guess. I don't know. And people have been interested in looking at them with ground proofing radar. So it's been something I was really interested in. And I worked with some people, not on that, but on other things. But they dug huge holes in the earth that, they were excavating the nest.
Yes.
They were really, really large.
Yeah, sometimes getting like the full colony is a bit of work. We really have to dig a hole like a 2 meters by 1 meter deep or something like that.
Yeah.
And then sometimes only to get like a teeny tiny ball of fungus with 20, 50 ants. But sometimes if it's like the leaf cutting ants, they have like gigantic nests of, was it like 20 meters diameter, sometimes even 30, going 10 meters deep. So to dig up that completely, that's practically impossible. So then you really just go for some fungus. So you dig in, you'll hit a chamber where there's the fungus present, and then take a sample with you.
Yeah. Yeah, so are you going to have to write another proposal then when your 5 years is up? Or?
Yes.
Okay.
And in the meantime, I'm trying to apply for permanent positions there as well, of course.
That is possible too.
Yes,
then.
yes.
Yeah.
It's actually part of my grant. It's the idea that through this grant, will get a more permanent position.
Is it through the Brazilian government or?
It's the local government of Sao Paulo.
Oh, okay.
FAPESP. And they have these relatively prestigious grants for young researchers to, as a startup, to create a new lab, a new line of research, in the hope that you are able to establish yourself as a permanent member.
You sound like you're doing that. You're getting results.
I'm doing my best.
Yeah.
Let's hope so.
Yeah, no, I really like insects and fungi. That's what I've worked on, but not those. I was interested, when I walk my dog, I see evidence of leaf-cutting bees. Do you have these in Brazil?
I'm not sure.
Yeah.
I only know of them, but I'm not sure where they are. I guess they are, because I think they're tropical, if I remember well.
Well, I know that they occur in Arizona because one of the MSA students had won a prize working with them. But I had never seen such a thing, because they cut out little crescents on the edges. It's really amazing. I'd never seen it before.
It's very similar to the leaf cutting ants themselves. They make these like half moon
Yeah, I've
cuts.
seen those in. I've worked some in Panama and Bear, Colorado Island, and there are tons of them there.
Yes,
Yeah,
yes.
yeah.
I've been there many times as well.
So great
Uh,
place.
Because my supervisor in Copenhagen, he had a very strong connection with Stry.
Yeah.
So we did all our field work in Panama. So I've been many
Okay.
times,
Oh.
mostly in Gamboa, but I
Yeah.
visited BCI several times.
Well, I'd rather work in because I'm afraid of snakes. So I was always happy to be a BCI.
And I've never seen a snake there.
At Gamboa, you never saw
I think
snakes?
only one, and that was, or maybe two, but both were non-poisonous snakes.
Oh, okay.
So everybody was always afraid of snakes, running and seeing them everywhere, and I never saw any.
Well,
I was like, where are they?
we did see one snake on BCI, but it was a non-poisonous snake. But I hear these horror stories.
Yes. But
Yeah.
I really enjoyed going there to do the field work. It's like rolling out of bed immediately in the field to do the collection. So yeah, it was amazing time.
It was really nice working on Bear, Colorado Island too. You're right there. We never got off the island, and we found over 300 undescribed yeast.
Oh, that's fantastic.
Many of them in the gut of insects, beetles.
Yeah.
Yeah.
Yeah, BCI is a beautiful place to visit.
So, so what direction are you going to go with your animals now? Your fungi and the.
So as I briefly mentioned, I work on the asexuality of the fungus. The idea is that the mutualism is stable because the fungus is maintained asexual. The ants, they transfer their fungus vertically, so the new queens, they always have like a piece of fungus in their mouth cavity, which they spit out when they create a new colony and the fungus start growing again. So for dispersal, the growth of mushrooms is not necessary. So if the fungus would start growing a mushroom, that's a lot of energy wasted for the mutualism as a whole. So somehow we can see that we barely ever find mushrooms in these colonies. The ants or the system is suppressing the growth of these mushrooms. So I'm looking at this from several different angles. The genetics in general, because the fungus of leaf cutting ants is polynucleate but also a polyploid. We have estimated about 6 to 7 different genomes within a single mycelium. And polyploidy often can affect in a
The reproduction.
in a way like the sexuality of a fungus or of an organism. I'm looking into mycoviruses. There are some mycoviruses well known in oyster mushrooms and in white button mushrooms that once they have infected the mycelium, people have problems growing mushrooms from them. They've been well studied because of economic interests. So I'm looking into seeing if there is any viruses involved in this as well to castrate the fungus, so to say.
Yeah.
And the third angle is how the ants themselves are actively changing this. Maybe if there is a fungus, they are actively transferring the viruses around. And the other option is they might be able to mechanically change the gene expression of the sexual genes in the mycelium.
So are the mating genes intact?
So that's one of the things that I'm trying to look into, but I haven't gone that far yet.
Oh, okay.
I'm in the start of this project. And I will compare it with the free living ones to see, like, is there a change indeed happening in the structure of those genes? Are they still viable, those genes, or not, compared to how it was before? So there's still lots of work to do.
Yeah, with some of our yeast that live in the gut of mushroom-feeding beetles, the mating genes are just shredded. You know, they're not intact. Although I'm told with yeast you might be able to get around having mating genes that work. So
yes.
But anyway, we've never been able to find in our mushroom-feeding beetles. We've never been able to find in one group any sex in the yeast. We've never found ascospores.
Yeah,
So,
because who knows? Maybe the beetles themselves are suppressing
Yeah, it
the
could be
expression
that
of those
too.
genes somehow.
Yeah, and then also with the mating genes being the way we find remnants of them sometimes, but some of them have none at all. Yeah, so it's really, really interesting. I know Cameron Curry.
Yes.
Have you run into Cameron working on some of these?
Once in Panama,
Yeah.
I ran into him in his lab. We've done some work together, also together with my colleague here.
Yeah.
There's many different groups actually working with these, but most of the people work on the ants and not on the fungus.
Yeah.
So I was able to find my niche to like really explore the fungus more.
Right. Right. No, it's really an interesting organism. Organisms.
Yes.
Yeah, and it's always been interesting that one group went off and with some of the ants. It's so different from your mushrooms. Yeah.
Exactly.
So. Okay, well, this is really interesting. I'm glad you could talk about this. And
fantastic. I'm glad to share this with you.
Yeah, yeah, and I hope you have good luck with your proposal.
Much.